We are accustomed to flattering ourselves with the illusion of omniscience, believing that the laws of physics and biology reliably protect us. But on our own planet, there is a place where terrestrial science literally breaks under the weight of the ocean. To understand the scale of this existential horror, we must look at how water behaves under extreme pressure. Under normal conditions, on the surface of the Earth, water molecules form a stable network resembling a tetrahedron. But if we descend into the deepest points of the Pacific Ocean, for example, into the Mariana Trench to a depth of 11 kilometres, the hydrostatic pressure will reach over 1,000 atmospheres, which is equivalent to a weight of 8 tons per square inch. Research conducted using the Advanced Analytical Facility ISIS neutron and muon source, which fires beams of neutrons at water samples, revealed a horrifying picture. Under pressure, hydrogen bonds distort, become less stable, and the network of water molecules itself compresses. Pressurized water forcefully breaks into the internal structures of proteins in living cells, destroying them and halting all vital biochemical processes. To survive, deep-sea fish use a chemical anchor, trimethylamine N-oxide, TMAO. This molecule acts as a piezolight and a chemical chaperone. It strengthens hydrogen bonds and prevents water from tearing proteins to pieces. The mathematics of the abyss are ruthless. The deeper a fish lives, the more TMAO it needs to accumulate. If shallow water fish have only 40 to 50 milliosmoles per kilogram of TMAO, then at a depth of 4,850 meters, this figure rises to 261 millimoles per kilogram. But this chemical armour creates a paradox that builds an invisible wall at the bottom of the ocean. The accumulation of TMAO inevitably increases the internal osmolality, concentration of dissolved substances, in the body of the fish. Most shallow water marine fish are hypoosmotic. Their internal osmolality is about 350 milliosmoles per kilogram, whereas the salty seawater around them has 1,100 milliosmoles per kilogram. The evolutionary history of bony fishes originates in fresh waters, where they were hyperosmotic relative to the environment and used kidneys with glomeruli to filter excess water. Upon transitioning to the sea, they deactivated or lost these glomeruli so as not to lose precious fluid. When a fish descends into the abyss, the level of TMAO artificially raises its internal concentration. Scientists caught a unique Hadal snailfish, Notoliparis kermodescensis, in the Kermadec Trench at a depth of 7,000 meters, which demonstrated record figures. The TMAO content in its muscles was 386 millimoles per kilogram, and the total osmolality reached 991 milliosmoles per kilogram. Extrapolation of these data indicates that at a depth of 8,200 to 8,400 meters, fish will become completely isosmotic with seawater, 1,100 milliosmoles per kilogram. To descend even deeper, they would have to accumulate so much TMAO that they would become hyperosmotic. For this, a fish would need to stop at a depth of 8,400 meters and wait for hours or days until its physiology reprogrammes and the kidneys start filtering water again, like in their freshwater ancestors. From a biochemical point of view, this is impossible. That is exactly why, below 8,400 meters, not a single fish exists in the world. It is an absolute physiological limit. Those species that managed to approach this limit of death closely were forced to dismantle their own anatomy at the genetic level. Meet the Mariana Hadal snailfish, Pseudoliparis swirei, an apex predator inhabiting depths of over 7,000 meters in the Mariana Trench. This creature is a perfect illustration of how evolution sacrifices everything for the sake of survival. The absence of light made vision and color a useless waste of energy. Genomic analysis of Pseudoliparus swiri showed that this species completely lost the pigmentation gene MC1R, turning into a transparent ghost. It also lost several important photoreceptor genes, retaining only rudimentary remnants of a visual system. But the most radical changes affected the skeleton. In conditions where a closed cranium would be instantly crushed by the pressure difference, nature found a twisted solution. Microcomputed tomography showed that the skull of this snailfish is not completely closed, and the majority of its bones are soft cartilages. This is no accident. Geneticists discovered a fatal frameshift mutation in the osteocalcin gene, BGLAP, which is responsible for tissue mineralization and skeletal development. This mutation, an insertion of a single nucleotide, causes the premature termination of cartilage calcification. Experiments on zebrafish embryos, where the BGLAP gene was blocked using specific antisense morpholino oligonucleotides, MO, confirmed that its deactivation radically reduces the mass of mineralized bone tissue. The snailfish permanently left its skull open so that external and internal pressures could equalize. Furthermore, under colossal pressure, the cellular membranes of ordinary living beings lose fluidity, becoming rigid, which blocks transport functions and leads to the death of cells. For membranes to remain liquid and elastic, they must contain many unsaturated fatty acids, such as docosahexaenoic acid, DHA. The final step of DHA biosynthesis is peroxisomal beta oxidation, where the key enzyme is acetyl-CoA acyltransferase, the ACAA1 gene. While ordinary fish have only 5 copies of this gene, the genome of the Mariana snailfish has expanded it to 15 copies. To prevent the denaturation of proteins and maintain extreme levels of the life-saving TMAO, the snailfish also has 5 copies of the FMO3 gene, which synthesizes TMAO. Moreover, one of the copies, FMO3A, has exactly 5 promoters for maximum expression. And finally, their key chaperone protein, HSP90, which is responsible for the correct folding of other proteins, received a unique point mutation, a substitution of alanine for serine near the ATP binding pocket. This microscopic structural adjustment acts as a mechanical lock that protects proteins from crumpling under the pressure of 1,000 atmospheres. When we cross the boundary of 8,400 meters and leave the fish behind, a new, invisible enemy awaits us. The ocean is not simply water. At a depth of over 4,000 to 5,000 meters lies the so-called carbonate compensation depth, CCD. Below this invisible line, colossal pressure, low temperatures, and a high concentration of dissolved carbon dioxide make the water so acidic and aggressive that it begins to act as a chemical solvent. It literally dissolves calcium carbonate, of which the shells and carapaces of marine creatures are composed. According to the logic of biochemistry, any crustacean that finds itself at the very bottom of the Challenger Deep, almost 11,000 meters, must instantly lose its carapace and dissolve alive. But it is exactly here that a tiny amphipod, slightly more than 3 centimeters in size, Hirondelia gigas, dwells. This creature found a way to outsmart the acid hell, transforming itself into a true knight in metallic armor. Seawater contains almost no aluminium, but Hirondelia gigas consumes plant debris and toxic mud from the bottom. With the help of specific sugar compounds in its gut, this amphipod extracts aluminium ions directly from the dirt that it swallows. In the alkaline environment of seawater, these aluminium ions turn into an aluminium hydroxide gel, a compound that humans use to protect stomach walls from acid. This metallic gel covers the entire exoskeleton of the amphipod, forming an absolutely impenetrable barrier. Aggressive water and high pressure simply cannot reach its calcium carapace. This is the first and only creature on the planet known to science that is capable of extracting metal from dirt to forge itself armour for protection against the ocean that is trying to digest it. We search for giant monsters at the bottom, but the true rulers of the Challenger Deep are creatures without a brain, without eyes, and without a nervous system. This isolated, extreme world is a hub of frantic microbial activity. Thanks to the V-shaped topography of the Mariana Trench, organic carbon slides down and accumulates at the very bottom, creating a nutrient medium for bacteria and archaea, such as proteobacteria, Planctomycetota, and Thormarchiota. But above these bacteria stands an invisible and ruthless puppeteer: Hadal viruses. During the investigation of 13 sediment cores from the Challenger Deep, at depths down to 11 kilometres, scientists identified 1,628 viral species, VOTs, previously unknown to science, belonging to 1,299 genera. The most terrifying thing about this figure is that 77% of these viral genera have never been encountered on Earth before. They are not in other oceans, nor in soils, nor in permafrost. This is genuine genetic dark matter, perfect biological machines that evolved in absolute darkness. Since there is no sun at a depth of 11 kilometers and food is critically scarce, these viruses ensure the functioning of the entire ecosystem through a mechanism known as the viral shunt. They massively infect and rupture the cells of host microbes, releasing up to 35% of all available labile carbon back into the ecosystem. The ruthless viral meat grinder literally feeds those who survived at the very bottom of the planet. Analysis of their genomes showed that lysogeny, when a virus sleeps in the host's DNA, occurs in only 5% of cases, which points to an extremely aggressive, lytic nature of their existence. But the viruses of the abyss do not just kill. They reprogram the anatomy of their victims. Hundreds of auxiliary metabolic genes, AMGs, have been found in their genomes. Viruses use these genes as a cybernetic code to force the infected cell to change its properties for the sake of the survival of the virus itself. For example, researchers discovered the genes cis-C and cis-H, which are responsible for assimilatory sulfate reduction. Viruses force bacteria to process sulfates, which helps host cells to create thiol compounds for the detoxification of heavy metals, such as arsenic and mercury, which accumulate in large quantities in the bottom mud. Even more striking are the viral genes responsible for the biosynthesis of lipopolysaccharides, gene LPXA/D and WAAE/F/L, and rhamnose, gene RMLB/C. The logic of this biomechanical hack is astonishing. The virus artificially thickens, strengthens, and cements the outer cellular membrane of the host bacterium with the help of these polysaccharides. It creates additional armour for the bacterium, so that it is simply not crushed by the hydrostatic pressure of over 1,000 atmospheres, while new viral copies assemble and multiply inside it. These entities have learned to manipulate foreign flesh to outsmart the deadly physics of the ocean. We leave the micro-world of viruses and return to the macroscopic horror that drove scientists mad for years. In October 2014, autonomous underwater hydrophones deployed near the Mariana Trench recorded an acoustic anomaly that did not fit into any framework of biological nature. This sound received the name Western Pacific Biotwang. It begins as a deep, low-frequency moan at a frequency of 38 hertz, a sound so low that you feel it more through the vibration of your body than with your ears. This introduction is characteristic of baleen whales. But what happens next breaks all the rules. The moan suddenly transitions into an extremely high-frequency, metallic, cybernetic twang at a frequency of 8,000 hertz. This finale sounds so artificial that researchers compared it to the sound effects of starships from Star Wars. It sounds as if a giant mechanism is trying to transmit a digital signal through the depths of the ocean. For almost 10 years, scientists could not identify the source of this sound. The solution came thanks to a combination of visual observations and advanced artificial intelligence. In 2018, researchers near the Mariana Archipelago observed Bryde's whales, Balaenoptera edeni, 10 times. And on 9 occasions, this eerie biotwang sounded simultaneously. To confirm this conclusively, scientists, using AI tools from Google, analyzed over 200,000 hours of historical acoustic data. Algorithms were trained to recognize the specific frequencies of this sound in the oceanic noise. The results showed that this is indeed the vocalization of the western North Pacific population of Bryde's whales. However, this answer generates a new existential chill. Why did an organic creature evolve to emit such a twisted, synthetic signal? The AI analysis also showed that the frequency of these sounds rose rapidly during the strong El Niño in 2016 and almost disappeared during the La Niña in 2021, which indicates a connection with changing climatic conditions and prey migration. Researchers hypothesize that this piercing metallic twang is their extreme contact call, a kind of desperate game of Marco Polo under the conditions of oppressive darkness and the boundless ocean. The ocean is so vast, isolated, and indifferent that its inhabitants had to turn their own voices into industrial sirens, simply so as not to be lost forever in this void, and to find each other over huge distances. Where our laws of physics and biology ought to have put a full stop, the abyss is only just beginning its cruel game. We have seen a world where water becomes a solvent that eats away bones, where fish are forced to live with open skulls and dissolve in their own chemical defense, and where tiny amphipods forge themselves metallic armour out of toxic mud. We have peered into the dark matter of the micro-world, ruled by invisible puppeteers, thousands of viruses unknown to science, manipulating the DNA of their victims, forcing them to build cellular shields against colossal pressure. And we have heard how lonely titans are forced to turn their voices into mechanical cries to overcome the absolute, endless isolation of the ocean. We are still only guests on our own planet. This deep-sea world operates by its own incomprehensible logic. It does not require our understanding, and it is absolutely indifferent to our presence. If you are ready to look truth in the eye and continue exploring the darkest corners of the Earth with us, leave your mark. Leave a like on this video. This is your signal into the void, which helps our project to break through the ruthless algorithms of the platform. Subscribe to the channel and make sure to press the bell. Our descents will only become deeper. And until the signal has disappeared completely, write in the comments, what causes you a greater existential horror? The invisible cybernetic empire of viruses that controls the bottom of the world, or the metallic cries of lonely titans echoing in absolute darkness?